It's my pleasure to welcome you to this virtual presentation on tissue cytometry, which is empowered by precision microscopy through artificial intelligence. And I would like today, in particular, focus on the applications in immunology. As Professor Henning already mentioned, I'm the Chief Executive Officer of a company named TissueGnostics, but I would also like to mention that in a side role, I have received an honorary invitation to serve as Adjunct Professor at the Queensland University of Technology, QUT, in Brisbane, in Australia. What is tissue cytometry, ladies and gentlemen? I assume you are all familiar with flow cytometry. Flow cytometry has been available for some 50 years. It is a method that allows to quantify antibody-mediated staining and detect the fluorescence signals on single cells, with one major limitation, that these cells have to be in suspension. That's why it's called flow cytometry. If you search on PubMed for FACS or for flow cytometry, you will literally find hundreds of thousands of papers that have been published over the decades. Tissue cytometry brings the idea of flow cytometry to the slide, and in particular to the tissue section, where we automatically identify single cells and quantify how much staining is associated with each nucleus. This works in bright field immunohistochemistry as well as in immunofluorescence. Like here, we can also detect multicellular structures and do what is called immunophenotyping or contextual tissue cytometry. But we can also look inside single cells and work with subcellular compartments, but also with pathogens like bacteria, viruses, or intracellular parasites. Professor Henning mentioned it already. The company was founded in 2003. That means next year we will celebrate our 20th anniversary. The global headquarters of my company, TissueGnostics, is still located at the place where it was founded in Vienna, in Austria, in Europe, in this beautiful building from 1890. But nowadays we have an international network of subsidiary and offices in Europe, in the United States, throughout Asia Pacific. I myself, as I said, I'm currently based in Brisbane, TissueGnostics Australia division, but we also have support offices and structures in South Africa, in Brazil, and in United Kingdom. As of this month, our list of reference papers has exceeded 2,200. This more than 2,200 research groups in more than 60 countries on all continents have published scientific papers in almost 800 peer-reviewed journals using instruments and or software from TissueGnostics. I would also like to mention that TissueGnostics is not only a software company, we are an ISO 13485 certified manufacturer of medical devices. The devices I'm referring to are called TissueFax. It is a microscope-based scanning system. The microscope comes from one of the best manufacturers in the world, Carl Zeiss. TissueGnostics is OEM partner of Carl Zeiss Germany, and the instrument is available in different configurations. For bright field scanning, fluorescence, this would be here in the center, is TissueFax Plus, our most frequently sold system, but we have different configurations available, like with a spinning disc, for a whole slide confocal imaging, an inverted system where you can also work with microwell plates to live cell imaging or time kinetic experiments. Then there is a high throughput system with a slide loader for 120 slides and a spectral imaging system with a liquid crystal tunable filter for multispectral imaging of at least 8 fluorescence colors simultaneously on the same slide. Like with any other slide scanner, the immediate result of TissueFax scanning process is a virtual slide. We see here an example of a prostate. We have here in red the secretory epithelial cells of the prostatic glands, in green a P16 positive basal epithelial cell layer, in orange the A marker, tumor marker, and we can see here how the tumor literally invades the normal adjacent tissue. This in green here, by the way, is a prostatic stone. And like with Google Earth, we can navigate through our sample and zoom in to see the details down to the cellular or subcellular details. This also works with cell cultures. We can see here a 5-color immunofluorescence of a neuronal cell culture in a well plate. And again, if we zoom in, we start seeing more and more details and faint structures down to subcellular details and subcellular co-expression patterns. As we support also spinning this confocal, we can also do whole slide confocal imaging. We see here an example from one of our clients in the US, Janelia Farm Research Campus from the Howard Hughes Medical Institute in Virginia. This is a section through an entire mouse brain. The tiny rectangles that you can see here are in fact not tiny. These are the individual high power fields of view that had to be acquired in order to capture the entire mouse brain section. There are 350 fields of view. Each contains 4 channels. And in total, scanning of the mouse brain section in a single focal plane at 20x, it took 8.5 minutes. If we, however, acquire also a set stack, so different focal planes, 21 to be exact, so the main focal plane, 10 layers above, 10 layers below, we acquire in total 29,400 images or 115 gigabytes of data for the single mouse brain set stack. And this is done in 1.5 hours, which is still very fast. If you try to do this manually, first it will not work, but if you try to, you will sit a week. Now, as I said, like with Google Earth, we can navigate through our sample, zoom in. Here, the map always shows us the overview of the entire sample. The red rectangle here indicates what is currently visible in the main window. And the more we zoom in, the more details and subcellular details become visible. But unlike other slide scanner companies, for TissueGnostics, the virtual slide is only an intermediary result, which we use to apply our algorithms upon and generate numerical data. So image analysis or tissue cytometry, to be more specific, is the ultimate goal. We have developed a set of algorithms that allow to automatically recognize single cells in tissue sections. And we typically use the nuclear marker as what we call a master channel. We can see here a standard immunohistochemistry peroxidase staining with a marker, well-known marker Ki67 in brown. And the obvious question is how many cells are proliferating, or in other words, how many blue nuclei are stained in brown. We made a survey among more than 200 scientists and asked them to estimate how many cells are positive here. So how many blue nuclei show a brown staining pattern in percent. And believe it or not, the estimations that we received from experts ranged from 1 to 40%, which I think is a major difference. Using our system, we use, as I said, the nucleus as a master channel, and the yellow contours here indicate what the software has identified as a single cell. And for each event, we measure how much blue and how much brown is there. And this is shown in such a scattergram, or people in flow cytometry call it a dot plot, where on the x-axis we see the hematoxylin mean intensity, and on the y-axis the Ki67 brown mean intensity. Each dot in here represents a single cell from the previous image in the tissue section. And this line here is well known from flow cytometry. It is the cutoff that has to be set in accordance to an isotype matched negative control. And only if cells which have a staining intensity greater than the value shown here, 20, will be considered as positive. And as you can see, the number of positive cells is 14.5%. And the important thing is, whenever you do this analysis on Monday morning, on Wednesday noon, on Friday afternoon, in Chile, in Australia, in Austria, you will always get the same result rather than a more or less arbitrary number. One of the features that our clients around the world appreciate most is the ability to track back and forth individual cells between the virtual slide and data space. You can always click on a cell in the image. This is, by the way, a renal allograft rejection. We see in green CD3 T cells and in red CD68 monocytes. And when you click on a cell, it starts blinking in the scattergram here. So you know exactly where this cell is located. When you click on a T cell, it's green positive, red negative. When you click on a monocyte, it's green negative, red positive. Following our motto, from image to data, we consider this a step forward and thus call it the forward connection. It also works the other way, from the data space back to the image. You can make a gate, which is a selection of a subpopulation of cells, like in flow cytometry. The cells will be shown in a certain color in other scattergrams, but they are also shown here in the image in red, in the virtual slide. So you know where a certain subpopulation of cells or certain phenotype is located in your tissue section, in your sample. So you have an immediate link between a cellular phenotype and histology. We call this backward connection or reverse gating. K67 is a marker that is expressed in the nucleus. And so we just identify the nucleus and use the same measure mask to measure also the brown staining. In case we have cytoplasmic marker, we use a secondary measure mask. It's also the nucleus, the primary mask, but we generate a secondary measure mask around each nucleus that captures the cytoplasm or the membrane stain. We see here an example of a four-color immunofluorescence staining from a client in Perth in Western Australia. It's DAPI in blue, T helper cells in yellow, cytotoxic T cells in red, and FOXP3 in green. You will probably know that CD4 FOXP3 double positive cells are referred to as the regulatory T cells. We use DAPI as master channel and then measure for each nucleus how much yellow and how much red and blue and green is associated with it, and get then this type of readout. You see here DAPI mean intensity versus CD4. So this gate here with backward connection in red shows the CD4 positive cells. Some are very strong positive, like this cell. This will be somewhere up here. Others have a lower intensity, like this one. This will be further down, closer to the cutoff, but all above the cutoff. And the cells which have a yellow staining below 65 will be shown here with a green contour. They are CD4 negative. Then this would be the analysis for CD8, and here finally CD4 FOXP3 double positive cells. Like in flow cytometry, we have two cutoffs in the scattergram. This is the cutoff for the parameter shown on the x-axis. This is the cutoff shown for the parameter on the y-axis. And these two cutoffs separate our diagram into four quadrants. The upper left, the upper right, the lower left, and the lower right. The upper right contains the double positive, the lower left, the double negative, and the upper left and the lower right, the single positive cells. It's exactly, you read this exactly like you would read a dot plot from a flow cytometer. This was both were examples of single cells. which were more or less round. I mean, the nuclei with Ki67 was very much round. The tissue infiltrating leukocytes are still quite round, but there are also cases where the cells are not round at all. And here we see an example. And I would like to emphasize that we have also algorithms that allow the segmentation of polymorph cells. Yeah? For example, here you see structures on the red, or here on the green cells in this staining. And this was, in fact, there's a nice publication from a group in Erlangen and Heidelberg in Germany, for whom we developed this solution to detect fibroblasts and dendritic cells. And you see in green, there are the fibroblasts, and in red, the dendritic cells. And they are anything but round. But still, also these quite strange or difficult structures can be identified. And then you apply these measure masks to measure all other markers which you might be interested in. But so this is just to show that with our tissue cytometry solutions, we do not request all cells to be round, because we know that not all cells are round, especially not in tissue. You might get round cells in a flow cytometer in solution, but not in situ in a tissue section. Quantification of cellular compartments is also an important application. I have here one example from Professor Henning's lab in Sao Paulo at University of Sao Paulo. Professor Henning, as you know, is on site, and he can tell you probably more than I can. But what I have understood is that this is an application, an example of activated microglia. It's a mouse model to work on Alzheimer's disease. And the question was here to determine the degree of fragmentation. So what we did is that we identified the cellular bodies shown here in yellow, and then in green, the fragments of the microglia cells. And in this scattergram, we see now 3 different populations of fragments. And please note that in this case, a dot here, an event in the scattergram, does not represent a single cell, but a fragment, a microglial fragment. And in orange, you see those fragments which are distant from the next cell body. Distant means further away than 50 micrometer. You see them here in orange. In red are those fragments which are attached to a cell body and not longer than 40 micrometer. And in blue, finally, the fragments which are attached to a cell body but longer than 50 micrometer. And this was in a project where they studied pharmacological effects, also effects of different pharmacological agents in mice, and on the wanted to determine the degree of fragmentation in the mouse brains of these activated microglia cells. In another example, which is also highly relevant from an immunological point of view, we have developed a special algorithm in collaboration with the Institute of Immunology in Regensburg in Germany, the lab of Professor Uwe Ritter. He's working with Leishmaniasis. This is an intracellular protozoan parasite that is transfected by mosquitoes. Infects certain cells like macrophages, and patients suffering from this disease really see severe structural tissue damage. I'm not going to talk about the disease as such, but about the methodology, how we can quantify intracellular parasites in situ and also in vitro. In Regensburg, they're using the inverted setup of our system, the TissueFax-I, where you can scan here micro-well plates, like from 6 well to 384 well. What we see here is a single well from a 24 well plate. And what we see here, the rectangles are the individual fields of view that had to be acquired in order to capture the entire well. In blue, Darby stain, we see the nuclei of macrophages. If we zoom in, then we see also the small dots here. These are the nuclei of the parasites. And after identifying the nuclei of the macrophages, we generate a secondary measure mask, which gives this ring here. That's why we call it a ring mask, where we can quantify the small dots, which are the parasites. The algorithm, of course, does not know what is a parasite. So we have to determine or support the algorithm in finding a threshold. So this is the control population without Leishmania major. All the dots here would be false positive. So we set here the algorithm, the sensitivity of the algorithm in a way that it doesn't find anything, and apply the same sample to the other. And then we get a nice detection of parasites per cell. TissueFax has also been used in a number of studies where a viral load or even individual viral molecules, viral particles had to be detected, like here in influenza, the viral load on lung parenchyma, or Zika virus infection, where individual virus molecules had been were counted in single cells. Dengue virus infection, which is a good example to show that with TissueFax you can not only analyze major cell populations, but also look for small populations like 0.5, 0.2, 0.09% of the cells positive for a marker. I mentioned that we have a system for spectral unmixing, and would not want to emphasize that this has been used in corona research, and a very prominent publication from Harvard University was published online in August 2020, so just a few months after the onset of the pandemic, where they have done a 7-color immunofluorescence staining, unmixing with our TissueFax spectral system and quantifying the markers with our software. And this was one of the first indications showing that the coronavirus tricks the immune system and avoids the formation of follicles with germinal centers, which have significant implications for the expectations of herd immunity. We know this nowadays, but please keep in mind that this paper was published in August 2020, when there were still governments who believed that they don't need to do anything against corona, but just wait for herd immunity to come. An approach which is very important nowadays to increase the number of markers that can be analyzed on the same population of cell on the same sample is multiplexing. There's a very nice protocol published from Kim Blenman at Yale University, how such multiplexing can be achieved using the TissueFax system for scanning and StrataQuest software for subsequent alignment of the individual staining rounds. Because in multiplexing, the same tissue section is stained multiple times with the same combination of fluorochromes, but in each staining round, the fluorochromes are conjugated to different antibodies. And these individual scans are aligned and through cross-correlation synchronized so that we can do co-expression analysis on 10, 15 markers on the subcellular level. And please look on how many markers are analyzed here. But two things are important to understand. Each dot that you see here represents a single cell, and all scattergrams that you see here come from the same tissue section through multiplexing. In our software, we have an extensive machine learning module, which allows to automatically classify structures in a tissue, like different immunological zones in lymphatic organs. And after applying a training, which the user can do within a few minutes, the settings, the model can be applied to an entire tissue section or a set of tissue sections, and then can be combined with the single cell analysis. And you will get the readout how many cells of which phenotype are in which of the histological classes that have been identified. So we have seen that we can scan bright field samples, fluorescence, confocal, multispectral, multiplexing, and single cells, subcellular structures, multicellular structures. We can now combine everything and do what we call contextual tissue cytometry. Others call it spatial immunophenotyping. We see here colorectal cancer, and dark blue is STAPI, in light blue cytokeratin, so the tumor area, in red CD4, in green FOXP3. We identify all the single cells, the tumor area is indicated here by this orange mask, and then combine the mask and can ask all kinds of questions, like how many CD4 positive cells are there, how many are in the tumor, how many are in the stroma. If they are in the stroma, how far are they away from the next tumor area, and how many are of the CD4 positive cells are also FOXP3 double positive. And in this scattergram, we see what is called an input gate on CD4. That means each dot that you see here represents a CD4 positive T lymphocyte in a tissue section. On the x-axis is the nuclear area, on the y-axis the distance in micrometer. And we can now make here gates that represent distance zones. CD4 positive cells that are up to 25 to 50, 50 to 100 micrometers away from the next tumor area, and so on. If we now make this backward connection or reverse gating on the first gate here, then we see in red all those CD4 positive T lymphocytes which are just outside the tumor, but not further away than 25 micrometer. And this would be the gate on this, the backward gating on the second, 25 to 50 micrometer, 50 to 100 micrometer. If we now go back to this scattergram, this is what we call the quadrant statistic. We see here in total there are 7,234 CD4 positive T cells in this tissue section. 2,267 thereof, for 31.3%, are in a distance zone of up to 25 micrometer. 1,593 or 22% in a distance zone between 25 to 50, and so on. And we see that there is a clear gradient as the number of cells drops with the distance. So we see clearly that there is not a stochastic distribution, but a targeted invasion of tissue infiltrating leukocytes targeted on the tumor. And all this, this type of analysis can be done in an observer-independent and reproducible way based on the analysis of single cells in situ. And with this, I would like to conclude my presentation. I hope I could show you that with TissueFax cytometry or tissue cytometry, we can support you to move from image to data and thereby increase observer independence and reproducibility. I would like to acknowledge our academic collaborators. TissueGnostics has been a member of currently 6 EU-funded Marie Curie innovative training networks. Each of this project was funded by the European Union with several million euro. I would also like to appreciate my collaborator at Queensland University, Jyotsna Batra, as we work on a project that we call the virtual histopathologist. I did not talk about this really, but it's about the whole story that I presented here is based on these collaborative efforts. And last but definitely not least, our scientific advisor, Henning Ulrich, from the Institute of Chemistry, University of Sao Paulo. With this, I would like to conclude my presentation. Please feel free to contact me if you have any questions later on, but also on site is Juan Carlos from Ursulab. This is our business partner in Chile. And I hope I could show you that with tissue cytometry, you can analyze cells, the fundamental unit of life, at the precision that inspires. Thank you very much for your attention.